In a previous video, we discussed how the backbone of DNA is composed of ribose or deoxyribose sugars that are linked together through phosphates via 3' 5' phosphodiester bonds. We also talked about the directionality of DNA, which is always going to be from a 5' terminus to a 3' terminus, and the directionality by default proceeds 5' to 3'. If we can make a phosphodiester bond like this, then we can make another, and we can just keep making them and making the DNA larger and larger. Notice something about this. The bases are not really involved with the backbone or the main chain of DNA. This is very much akin to the side chains of amino acids that are not involved with the peptide bond. It's time for us to move on and talk in more detail about how the next level of structure is going to be thought of and composed. So we'll move on to the next page and talk about what I like to refer to as DNA secondary structure. That's a nice way to put it because it's parallel to the next level of protein structure we did, in which we looked at helices and sheets once we had done putting together the main chain of proteins. Similar situation here, and you may know that DNA's next level of structure is 3 dimensions and forms a double helix. So it is in some ways like the alpha helix, but different in many ways, as we'll see going through here. It is a double helix. It's 2 strands, and interestingly enough, it has what we call an anti-parallel nature to it, which is an idea we first introduced with beta sheets. That is, one of the strands proceeds in one direction, and the other strand proceeds in the other direction, and we have that anti-parallel directionality for these 2 strands. So the first thing we're going to do is lay this out flat as if it was not a helix, even though it in fact is. Okay, let's recollect what I just said, and that is that the bases stick out from the backbone, and you might have one that is a little larger. That would be a purine, and we might have some that are smaller that are pyrimidines, and these can be in any particular order that we want, and they are going to reach out and interact with the other strand potentially. Now on the other side, let's just use a different color just to keep this straight. If we have a purine on one side, we're going to have a pyrimidine on the other, and vice versa. More on this on our next video. Now, these bases are sticking out and interacting with each other. They're drawn as sticks because we're looking edge on to those. It's as if the bases are nice and flat, so we can't see that aromatic nature of them. Let's make very clear, though, that they are interacting with each other, and they do so with hydrogen bond donors and acceptors. More about donors and acceptors, and we're going to look at those again on the next page, something we'll see pretty much right through the final exam. However, this is something you might know from your general biology classes, but there's much more to the chemistry of how DNA sticks together. And I just want to touch on that very briefly here. We could point out that because these are aromatic rings, they actually have pi electrons that are forming clouds above and below the ring here. I'll just show them as these little green blobs, or we might have blobs coming off from here as well. And these pi electrons are on both sides of the ring. I won't draw them everywhere, but I would note that they interact with each other through what we call stacking forces. Let's make a note of that. Stacking forces are overlapping pi electron weak interactions. Notice that some stacking forces are between 2 bases on the same strand, and other stacking forces are between bases that are on opposite strands. What that means is that they can actually help hold the 2 strands together. So it's a little more complicated than we have talked about in your high school biology classes. Let's note that every base has a negative charge associated with it, That's on the phosphate between the bases, like that. And we can do that on both sides. And that means DNA is highly negatively charged, and we would typically expect to have some balance for that. If we do that in water, it's often magnesium ions, magnesium +2, able to fit right between two negative charges like that and balance them. And that's going to be an important counter force in cells and in water. More about that a little bit later. Now, this is about all we can do in 2 dimensions. At this point, we have to twist the molecules up into helix. And when we do so, we still have anti-parallel directionality, and we can follow those through and show that it's anti-parallel. We still have all of the same things we talked about just now. That is hydrogen bonds between purines and pyrimidines, and stacking forces. We still have negative charges, lots and lots of negative charges up and down here. I won't go ahead and draw them all in, but you get the idea. We can say a few more things about this, though. First of all, we can point out that it has very regular structure. In fact, maybe we'll move down here a little bit and make a note that the width of DNA is always 20 angstroms. Angstrom is a capital A with a little circle on top of it. That's about an angstrom is about 1/10 of a nanometer. So this is a very small molecule, but nonetheless, it's a regular width. It's got a very common width as you go along the DNA structure. The other thing we want to point out is that it is what we would call right-handed, like an alpha helix. That is, it has the same screw sense that a normal righty-tighty, lefty-loosey wood screw would have that you'd use a screwdriver for. It's not terribly important, to be honest with you, but it is a fact that we note. And there is one more area that's really important that I want to discuss, and that is the grooves that are formed between the strands as you twist these things up. So what I'd like to do is point out that the strands are not evenly twisted upon each other. In fact, the space between the strands here is fairly narrow, whereas the space between the next 2 strands is very wide. And what we're going to do is color these in to show you a little bit about these grooves. This large groove, which is between these 2 strands and actually goes way into the DNA structure to the back wall there, this is called the major groove for obvious reasons. It is the larger of the 2 grooves. It's very wide, it's very deep, and there's actually space in there that we'll take a look at in some structures. The smaller area between the other portion up here, this strand here, as you might expect, is called the minor groove. And that is obviously more narrow, but it's also more shallow. Nonetheless, there is a space there, and it is something that we need to be able to recognize. Now, one thing we often see people get mixed up on is, because of 2 dimension in our slide here, we sometimes think of the major groove as a triangle wedge, and it certainly is. You can see it right there, and maybe we would look at it down here. But keep in mind, it's all of this area, and in fact, the major groove comes up the back here behind the minor groove and out to here again. In fact, the major groove is a helix itself. It's a helical groove, and there's only 1 helix of the major groove for the entire molecule. Same thing for the minor groove. The minor groove you can see here comes across the front, but then it goes down the back here and then comes around to the front again. So the major and the minor groove are both helical grooves, and there's only 1 of each in an entire molecule of DNA. We're going to look at the major and the minor groove in a different way on the next page or the next video, and it's an important area because the structure of DNA and these differences are going to play a role in their function.